In the previous lecture, we talked about the steps of translation and how a ribosome machinery is put together and how it elongates the peptide, the polypeptide. And in this lecture, we are then now look a little bit into the details, how prokaryotes and eukaryotes handle the protein synthesis. First, a quick recap of the transcription itself. So we saw that there is an easy and a tricky transcription. Obviously, we saw that both are really not easy, but comparatively, the prokaryotic transcription was straightforward. So the transcript was made, the mRNA was ready to go for the protein synthesis, and everything happens, if you will, in the same room. In the eukaryotic cell, we are now in the nucleus, and when the transcript is made, this is definitely not ready to go. We had several steps to process this primary RNA transcript. Number one, it has to be spliced. The exons need to be put together. After that, we get the 5' capping, and at the 3' end, we got the polyadenylation. Only after these steps have been completed, the mRNA is ready to go and is transported out of the nucleus in order to get translated. So let's take a closer look at the mRNA transcripts that are ready to go. So even at this stage, we have big differences between the prokaryotic mRNA and the eukaryotic mRNA. Here you see that in the prokaryotic mRNA, we have in this example, 3 start codons lined up versus the eukaryotic mRNA with 1 start sign. What does it mean? The prokaryotic mRNA is polycistronic, meaning in one mRNA transcript, it will code for different proteins. This is very different in a eukaryotic cell. In vast majority of cases, there is going to be one protein to be translated and period. So let's take a look what happens with this polycistronic mRNA with many protein coded on one transcript. Well, in the prokaryotic cell, when the transcription begins and the mRNA emerges, which is also ready to go, the protein machinery already starts translating the transcript. So this is basically happening more or less simultaneously. Very different in eukaryotic cells. So here we indeed have a separation between the transcription and the translation. So the translation only happens in the cytoplasm after the transcripts have been transported out of the nucleus. But also a eukaryotic cell can then assemble many ribosomes on the transcript in order to make the protein more efficiently. On this slide, we take a closer look at the polyribosomes in a prokaryotic cell. So you see here the RNA polymerase making the transcript from the DNA, shown here in blue. And as soon as the transcript emerges, the ribosomes can start accumulating on this transcript and start making the protein. This is, in fact, one of the most fascinating things you can see under the electron microscope. So this was now the example in the prokaryotic cell. So in the next slide, I'm going to show you the visualization how this actually happens in a eukaryotic cell.
As soon as a messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously. Each ribosome begins at the 5' end of the mRNA and progresses steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When a ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.
We just saw that the prokaryotic mRNA has many protein genes lined up one after the other. And now the question is, how does the translation machinery find these start codons, the AUGs? So now here you see immediately before the start codon, this abbreviation SD. Well, this probably already fired a standard deviation in your brain. But here, this is actually another word we can learn. So this is this Shine-Dalgarno sequence. And the trick here is this sequence that is already in the mRNA, the matching sequence to that is also present in the small subunit in the rRNA, meaning the whole machinery can now position itself in frame with the methionine to start the synthesis. And I would like to also draw your attention to all these factors. So obviously we are going to need factors, initiation factors. In this case, we have 3. And if you think about the prokaryotes, and they already have these 3 different initiation factors, you can already imagine how many we are going to have when we look at the eukaryotic cell. Once the small subunit has positioned itself in frame with the help of the initiation factors and the Shine-Dalgarno sequence, the large subunit can now come in and complete the machinery. Here, the initiation factors will dissociate, and as soon as the large and the small subunit combine, we are going to then create these 3 different sites that we looked at in the previous lecture, the A site where the tRNA will come in, the P site, the peptidyl site where the synthesis will occur, and lastly the E site, the exit site where the discharged tRNA can then fly out of the machinery. The translation initiation in our cells is quite a bit different. So we do not have any of these Shine-Dalgarno sequences or anything. So here we are going to have to do with a lot of initiation factors and also the recognition of the mRNA. Remember, we have this 5' cap that actually also identifies this RNA as an mRNA, and this is what the small subunits then eventually recognizes. And with this initiator, methionine now needs to find this AUG, this start sign. In vast majority of cases, the machinery will then scan through the mRNA transcript in order to find the first methionine, the first AUG, to initiate the synthesis. Once the small subunit places the initiator tRNA with its methionine onto the start codon AUG, now the large subunit can come in and initiate the sequence. As you can see, the concept is indeed the same. So we have this small subunit with this initiator methionine that somehow finds this AUG. So this is different, but the concept is the same. And once this step is accomplished, the large subunit comes in and starts the synthesis. This slide gives you an idea about the complexity of the translation initiation in eukaryotes. So here we have way more factors involved in order to basically kick off this process in eukaryotes when we compare it to the prokaryotes. On this slide, we have the simplified schematics of the elongation step. So we know so far that the tRNA has to come in, the peptide bond has to be formed, the whole machinery has to translocate in order to walk through the mRNA, and the empty tRNA has to dissociate. Well, now the question is, what kind of picture do we have so far in order to understand the underlying mechanics of these complicated steps? One particularly important step during protein synthesis is to recognize the correct tRNA. Recall, the DNA polymerase was able to correct its mistake. When it had incorporated the wrong base, it could take a step back, excise the wrong one, put in the correct one, and continue. So ribosome does not have any of these abilities, so it has to make sure that the tRNA, the newly incoming tRNA, is indeed the correct one. And structural biology already gave us clues how this could happen. So here we see the structure of the rRNA in the small subunit, and we see these universally conserved nucleotides in the absence of the tRNA. And when we have the tRNA that fits the mRNA correctly, we have these huge conformational changes of these universally conserved nucleotides, and this appears to be a signal for the machinery that the correct tRNA has come in. So this is one step. The second step is, of course, how is it that this machine can catalyze this peptide bond formation? In order to get an idea what might be going on here, we go back to this clover leaf depiction of the tRNA. So you remember this universally conserved ending with this CCA at the 3' end. So the A is where this amino acid coupling takes place. So now the question is, if you then picture this ribosome, now you have one tRNA in the peptidyl side and the other that just came in the A side, charged with their respective amino acids. How do these two amino acids now form this peptide bond? So here the structural biology gives us some clues. So the reaction itself, as we saw at the very beginning, is really easy. So we have this nucleophilic attack of the nitrogen here from the incoming tRNA shown in pink onto the carbon atom of the tRNA that is in the peptidyl position. And the clue here is the positioning of these universally conserved CCs at the very end of the tRNA. So we see that these CCs are pairing with the GGs on the so-called A and P loop. So those are the rRNA components of the ribosome. So here the idea is that the job of the ribosome is to get everything into place using these universally conserved nucleotides in order to catalyze this reaction. And now we can appreciate why these specific nucleotides are kept conserved. We also have to end the translation once this machinery walks by one of the 3 stop codons. And we said that this stop codons are not recognized by any tRNAs. Instead, release factors come in to basically release the polypeptide chain and disassemble the whole machinery. So again, structural biology gives us some clues how this might happen. So this is in blue ribbon, you see the release factor, the crystal structure of the release factor that is superimposed on a tRNA. The idea is that this release factor basically mimics the tRNA. So this is a protein that mimics the tRNA, but instead of bringing in the new amino acid to be incorporated into the growing polypeptide chain, it just basically blocks the A site. So on the right, you see the superimposition of this release factor in the A site. We have now learned quite a bit about the protein synthesis machinery. So is there anything that we can then use from all this what we have learned in order to benefit the lab work. So recall that we have been able to make a DNA polymer very easily in the test tube. The only things that we needed to put in were the enzyme, the Taq polymerase, the DNA primers, and the letters. This was it. So the question is whether or not we can do anything similar like this with the protein polymer. It turns out no. So this would be the machine itself, and those would be the factors that we would need in order to get eukaryotic protein made in vitro. So this is definitely not going to help us a lot. So meaning we are going to have to ask a real organism to make the protein for us. So, and there because we have learned quite a bit from the translation machinery, we can then start designing our own protein. Maybe the two most important words in protein design are the start and the stop codons. So we saw that the translation machinery has found a way to specifically recognize the start of the protein and the end of the protein. That means these words, these codons, they have to be put in somehow in order to basically tell the organism, whichever we want to use, to start there and to stop there. Okay, how do we deal with that? So we go back to our vector that we have put together with everything that we have learned during these lectures. So we learned that we have to put in an origin of replication in order to convince E. coli, for instance, to make also our plasmid. We have learned that we should also have an antibiotic resistance to force this organism to keep our vector. We also learned that we should put a promoter in front of the gene so that this can be actually transcribed by the transcription machinery. Now we have to give the start and the stop sign for the translation machinery, and this we do at the DNA level, of course, by including the start word and the stop word into the primers before we do the PCR. Once the PCR reaction is done, the piece of gene that we have will then, of course, have the start codon at the beginning and the stop codon at the end. We also learned that we have to keep the reading frame because the genetic code will be read in triplets. So we are not allowed to have any frame shift because this is going to change everything. So let's look at this particular sequence. This is a sequence that will be recognized by an endonuclease. So it will cut this particular sequence. So let's say for some reason you have to include this in the middle of your gene. When you then look at the number of nucleotides we have here, then you will see that we have 3, 6, 7, 8 nucleotides. So meaning we have the first triplet, the second triplet, the last triplet is missing one nucleotide. So if we would just insert this particular sequence in the middle of our gene, this would then introduce a frame shift. And after that, whatever is read would be gibberish. So what would we do? We would then put another nucleotide in order to complete this 8 to 9 to keep the reading frame. Here we can then see what happens if the frames have been really shifted. So in number 1, we see the triplets with the CUC. The start is CUC. And the second one, not the CUC, but UC and then A. And you see already that this frame shift will then result in a completely different polypeptide chain than you wanted to have originally. Let's think about this restriction site we wanted to put into the gene. So this would then basically introduce a hole in the triplet code, and we would completely shift the reading frame. So basically what this tells us, never lose a base. We have to solve now one last problem. Let's say we have done everything right. We used all the words that we have learned and we convinced E. coli to make our protein. Now the question is, how do I fish out this protein in order to have it in my hands? So you burst open the E. coli. So your protein is swimming somewhere there, but also everything else. So what we are looking for is a fishing rod. How do we fish out this particular protein? And there we have again learned a lot from nature. In this particular case from influenza virus. So the example given here is the flag tag. So those are a couple of amino acids that can be recognized very specifically by a corresponding antibody. So we basically put this antibody onto our fishing rod. When we burst open everything, we fish out our protein, meaning we should better put this sequence onto our protein before we start all these procedures. So in addition to putting in the start and the stop codons, we also have to include the sequences that code for these couple of amino acids for this flag tag. So with that, we have basically learned from nature how we can start out with 4 letters and end up having our protein in our hands. It is indeed truly amazing how nature can use 4 letters in order to, let's say, write whole stories. So we are basically made of 4 letters. E. coli is made of 4 letters. But as we have learned at the very beginning, this really took us a while to understand how this works. So just realize that we have seriously managed to conduct war in the air with planes before we figured out that DNA is the hereditary material. And with that, I am leaving you with this take-home message.